I'm with Translatum Medicus Inc., or TMI, and TMI is a spin-off company from the University of Toronto where I'm an ophthalmologist and scientist. I'm a retina specialist. And for those of you who don't know me, I was the head of the ocular angiogenesis research program worldwide with Novartis during the initial due diligence in licensing and early development of ranibizumab. And I am founder of TMI. So we all know that age-related macular degeneration is the leading cause of blindness in the developed world, and unfortunately for the majority of our patients, there are no treatments. So with that tremendous unmet need, Translatum, in trying to efficiently translate science into medicine, focused directly on the major challenges. And those are that it's a complex disease of innate immunity. No animal models actually developed dry AMD. Clinical trial design is complex, and sustained release will ultimately be required. But thankfully, the quantification of GA is now a part of clinical trial design, and this is a patient of mine, and so we can measure and quantify those patches. And as you now know, clinical trial endpoints actually look at patch expansion and whether or not you can slow the rate of expansion. So TMI's approach is quite different for most people, and instead of focusing on the complement arm of the innate immune system, we actually focus on the cellular arm, and in particular on macrophages. And within macrophage biology, there's a simple hypothesis which sort of says there's good guys and bad guys. The good guys are your M2 macrophages, and these cells will gobble up the odd dead and dying cell without inciting inflammation. By contrast, M1 macrophages are very aggressive. I call these the dirty splinter macrophages. If you get a dirty splinter in your finger, they will chew up the splinter, the macrophages, and the tip of your finger. It turns out these macrophages are identified in tissue, and there's about 3 reports out there, including one from Chi-Chow Chan's lab at the NIH. So our hypothesis, therefore, is that we would want to turn an M1 macrophage back towards a neutral or an M2 macrophage. So I'm going to show you now how TMI has de-risked our drug development plan. So first I'm going to show you we created an animal model. And on the left you can see a patient with a patch of geographic atrophy quantified in blue. On the right, you can see an animal, one of our rodents that's actually developed geographic atrophy. And with a 1.5 years' work, we can now make those patches expand, thus aligning with clinical trial. Importantly, in this model, we learned that macrophages are important. And in this photo, which I quite love, you can see the RPE cells in yellow. They're sick and dying, and insinuating themselves between them are the macrophages in red. When we look for M1 markers, we can see them in those macrophages. And in fact, when we look at clusters of genes, we can see that they all sort of tip towards M1 polarity during active disease. Now I'll tell you about our lead compound, and it's our TMI-18, and it's a first-in-class transcriptional modulator. So instead of hitting a receptor or its ligand, it actually gets into the nucleus and changes messenger RNA expression. And in the upper panel here, you can see during non-inflammatory conditions that messenger RNA is actually driven from some of our key promoters at very low levels. During inflammation, so during the M1 response, you get more messenger RNA produced. And our drug can actually drop that down. And we've confirmed that, as well as others have confirmed that in our macrophage population. So the question is, what happens if you put it in our model? So what I'm showing you here on the left is an animal that's received saline, and you can see they get a large patch of geographic atrophy-like tissue loss, whereas when we give our drug, they dose-dependently get smaller patches. We can show that not only do we protect structure, but we protect function on the ERG, and when we take out the tissue, we see that we protect RPE cells. What I'm showing you here are 2 of our key targets. These are MCP1 and MCP3, and we can see the messenger RNA going down in our animals. And interesting, and we're very excited about this, many of the downstream mediators of inflammation, including the inflammasome, go down, including NLRP3. And so when you look at our cluster of genes, you can again see the messenger RNA shifting. And we're delighted with this. When we put TMI-18 into our patch expansion model, we can completely block patch expansion, thus aligning with clinical trial endpoints. So the exciting thing for us right now is TMI-18 has already been shown to be safe in people. It's been given in over 400 patients orally in phase 2 for autoimmune disease. Not only do we know its mechanism, but those studies confirmed it's got druggable targets and that it's scalable for GMP production. We've also confirmed with preliminary in vitro work that because it's a low molecular weight synthetic, it's entirely suitable for sustained release. And I'm delighted to tell you it's patent protected for ophthalmic indications. And although I am a spinoff from a university, I am the owner of the IP, which I have assigned to the company. So in summary, this is how we've de-risked our drug development plan. We have an animal model that uniquely mimics disease. We have a lead compound that's first in class, dose-dependently efficacious, systemically safe, scalable, and patent protected. And so now we're poised to accelerate. And so we, pending ocular toxicity studies and IND approval from the US FDA, hope to be into clinic very soon. And I'm also delighted that 11 days ago, we raised our seed round. We just closed our first round of investment, and we're already in discussion for ongoing investment. And finally, I'm delighted to report, so for those of you who saw me up here last year, we were a company of one, and I apologize for not returning your emails. We now actually have a company, and I've recently brought on Dr. Wayne Schnarr as our Chief Business Officer, and he was formerly VP of Oncolytics and Equicom. Mike Gray of Papa's Venture, formerly VP of Glaxo, has been a strategic advisor. And although we have not formed our committees or our boards, I've been had significant support from Dr. David Boyer, whom you all know, Julia Levy, who is former CEO of QLT, and Dr. Barrett Katz, who was with Fovea and Danube and is Director of Clinical Trials at Albert Einstein. So thank you very much.